You're about to hear a rebroadcast of Where We Live. It was originally recorded on December 21st, 2017.
This is Where We Live. I'm Lucy Nalpathanchel. Oceans make up 99% of living space on planet Earth. Today we hear from the scientists who study deep-sea creatures that make and re-emit light. Coming up, we'll learn more about biofluorescence and how a particular glowing gene from a type of jellyfish has led to advances in studying human genetics today. A Vanderbilt University biologist will also join us to explain how his lab is taking proteins from fireflies to better understand circadian rhythm. But first, what is bioluminescence? And why is this such a common trait among many sea creatures? In studio with me today is Dr. Mark Zimmer, professor of chemistry at Connecticut College in New London, author of several books including Illuminating Disease and Introduction to Green Fluorescent Proteins. Mark, welcome to the show.
Thank you.
I asked a question about bioluminescence. Can you briefly describe this for us?
It's any animal, living animal that's giving off light to attract prey, to scare away something, or to find food, communicate with other species.
On land, we're familiar with lightning bugs or fireflies, but actually a lot of creatures in our oceans use this or have this chemical reaction in them. Can you describe why?
Well, the deeper you go, the less light you get. And then the best way of communicating is by light. If you think about it, if you've got a firefly in a football field, far away, during the day you can't see it, but at night that tiny little flash, which only comes from about a quarter of its body, and we can see it very, very clearly. The same as stars. We can't see stars in the sky right now, but at night, as soon as it's dark, we can see the light. And so it's a very efficient way of communicating or finding things.
Now, someone who knows what it's like to be deep in the ocean is on the phone with us, Dr. Edie Witter, CEO and Senior Scientist of the Ocean Research and Conservation Association. Edie, welcome to the show.
It's very nice to be here.
You're a deep-sea biologist, so when you go down there in these robotic vehicles called submersible, what's that like?
Oh, it's just about the best light show you can ever imagine. My favorite place to be is 3,000 feet in the ocean near the end of a dive when we turn out the lights, and we're going to be coming up and just seeing these explosions of living light all around the submersible. It's spectacular.
I was talking with Mark in studio about the many creatures that are luminescent. Why is this, and how many are we talking about?
In the ocean, you're talking about trillions and trillions and trillions. It's just an absolutely astonishing number. The most abundant vertebrate on the planet is a little fish that makes light. And it's everywhere in the ocean because it's so vital for their survival, for helping them, as Mark said, find food, attract mates, and defend against predators.
When we talk about these sea creatures that glow, describe that for us. You were talking about what it's like to be down in the submersible, but when you're talking about glowing, what are you seeing exactly?
So most of it is blue. That's the color that travels best through seawater. And so animals have evolved the color that's going to travel furthest. But it actually can come in all colors, red, orange, yellow, green, blue, and violet. And so there are rare instances of other colors. But most of the time in the submersible, what I'm seeing is explosions of blue light. And sometimes they're just flashes, almost like a firefly's flashes. But other times they really are explosions of the release of bioluminescent chemicals into the water, sometimes as what looks like smoke, sometimes as particles, and sometimes, we'll see very clear outlines of jellyfish. There are these super long chains of a kind of jellyfish called siphonophores that can be meters and meters long, and sometimes we'll catch one on the submersible, and it arches in front of the sphere and forms this parabola of light glowing that looks like Japanese lanterns, and then it'll break and flutter in the current. It's just incredible.
In your research, can we find out a little bit more about why these animals at different points are using this light?
So one of the most common reasons for using it is camouflage. as you indicated in your opening, we're talking about most of the biosphere on the planet. Well over 99% of the living space on the planet is the open ocean environment. And this is a place where there's no hiding places. There's no trees or bushes or hidey holes. And yet animals have to play all the same games of hide and seek that they do on land. Predators need to be able to sneak up and prey, and more importantly, prey need to be able to hide from predators. And so as the ocean filled up with swifter and nastier predators, the only place to hide was to go deeper where it's dark. And so a lot of animals started to migrate into the dark depths. Trouble is, all the food's at the surface where photosynthesis occurs. And so a lot of these animals spend their lives going between the surface at night when it's safe to feed and then hiding in the dark depths during the day. And in order to be able to avoid being seen, they camouflage their silhouettes. So they produce light from their bellies that exactly matches the color and the intensity of sunlight penetrating from the surface. So if a cloud goes over the sun and dims the sunlight, they dim their belly lights. And it's spectacular. It's the perfect cloaking device.
We heard from Mark earlier talking a little bit about fireflies. This is one of the few terrestrial animals that actually can glow. And is that because we have this natural light, this ambient light, and more places to hide on land?
That's right. Presumably, as animals evolved out of the ocean and onto land, many of the early inhabitants weren't luminescent, and even if they were, they didn't have the need for it, and so there were plenty of hiding places on land, and so there hasn't been anywhere near the selective pressure that there is in the ocean to develop this enhanced form of visual communication.
I'm speaking with Dr. Edie Witter, CEO and Senior Scientist of the Ocean Research and Conservation Association. I should mention you're also a MacArthur Genius Grant recipient back in 2006. As we learn about bioluminescence and why so many sea creatures, more than 3/4, actually are able to use light that they create themselves deep in the ocean. In studio with me, Dr. Mark Zimmer, Professor of Chemistry at Connecticut College in New London and author of several books, including Illuminating Disease and Introduction to Green Fluorescent Proteins. And when we think about going someplace very dark, very deep in the ocean, Edie, I guess our inclination is to put on bright lights, but that actually would, I understand from your research, you found that doing that actually scares away these animals. You found a way to bring them to your submersible. Can you walk us through that?
Yeah, so the first time I went down in a submersible and turned out the lights and just saw how much light there was being made down there, I really wanted to understand better what it was being used for and what it was communicating. And so over the years, I've tried to imitate some of those displays to see who might respond to it. And I quickly realized that you had to be pretty sophisticated about this because the animals are easily spooked. And so I needed to be able to see without being seen, and so I used a red light that they couldn't see to be able to see them, and then imitated certain displays. And I found one in particular that imitated a certain type of bioluminescent jellyfish, seemed especially attractive to squid, and in fact so attractive that we eventually were able to use that imitation jellyfish, electronic jellyfish as we called it, to attract the first giant squid ever caught on film in its natural habitat.
I remember here reading about that, but I think it was in 2013, 2012, 2013. What was that like to see this giant squid? Explain to our listeners how big we're talking and how it looked different compared to, say, a giant squid that may have washed up and fishermen may have found it.
Well, this was not one of the bigger ones. Had its feeding tentacles fully extended, it would have been as tall as a 2-story house. But giant squid have been found as tall as a 4-story house. An awful lot of that is these super long tentacles that they have. But the thing that was most surprising about this was its color. Because when we see them washed up on beaches, they tend to be brownish, reddish color. And an awful lot of squid that I've seen from submersibles actually have that reddish tinge because it's a type of camouflage in the deep sea. But the giant squid had this amazing bronze, silver color. It would switch back and forth between bronze and silver, and it looked like it was carved from metal. It was incredible. And then this enormous eye, the size of a soccer ball, looking at you, with you couldn't help feel some intelligence. It was pretty amazing.
Well, I want to thank Dr. Edie Witter, a biologist, CEO and senior scientist of the Ocean Research and Conservation Association. We're going to tweet out a link to one of your TED Talks, Edie. I understand you describe yourself as a bioluminescence junkie, so we appreciate you coming on today to describe this phenomenon to us as well. Thank you so much.
Very happy to do it. Thank you, Lucy.
In studio with me is Dr. Mark Zimmer, a chemistry professor at Connecticut College. He's going to stay with us as we continue to talk about bioluminescence. And coming up, we're also going to learn about biofluorescence. How does the study of organisms that produce or re-emit light help scientists better understand what's going on inside of cells, including our own? We'll find out after the break. And find us on Facebook and Twitter at Where We Live. This is Where We Live. I'm Lucy Nalpathanchel. Today we're learning about why living organisms make and re-emit light. Research into this common trait, especially among animals in the sea, is helping scientists understand human biology. In studio with me, Dr. Mark Zimmer, professor of chemistry at Connecticut College in New London, author of several books, including Illuminating Disease and Introduction to Green Fluorescent Proteins. We're going to learn about what green fluorescent proteins are in just a little bit, Mark. But because we were talking about luminescence in the ocean and then fireflies on land, how is it different, the way they make this light?
So many of the organisms in the ocean are using their light to disguise themselves. The fireflies, we see them all the time. It's a big beacon. It's like saying, here, come and catch me. And it's only the males that are flying. They're sending a signal to the female, and the female sits hiding at the bottom. And when she sees a male she likes, she'll send a signal, come hither signal, and the male will fly down. If you next spring catch a firefly, shake it, you'll have caught the male because that's really the only one you see, and then lick your hand. It'll taste disgusting. And it turns out the males make this lucipophagon, it's called, and so bats don't catch them and birds don't catch them. There's one species of firefly where the female, once she's attracted a male, will attract a male of a different species, and then she'll eat him, spit out everything but this repellent. And she's the only female species, if you catch the female, who can also give this oozy, disgusting smell or tasting stuff.
And that also protects her eggs?
It protects her from being eaten because she does have to flash a light very briefly. Nobody really knew why she ate the male because while they're in the flying stage, they don't eat. And so why would she actually attract a male from another species and eat that male? And it turns out it's just to get this repellent and to defend herself.
Speaking of fireflies, our next guest does research using this protein from fireflies. Dr. Carl Johnson, professor of biological sciences at Vanderbilt University. Carl, welcome to the show.
Hi, thanks for having me on.
So we were talking about sea creatures that are bioluminescent, and you actually focus on a gene in the firefly in the work that you're doing in your lab. Why use these firefly proteins?
There's actually a lot of different organisms that bioluminesce. There's fireflies. There's also a number of jellyfish, different types of jellyfishes, and bacteria. And many of the genes that are responsible for this glow, called luciferase, the genes that make this protein called luciferase, have been isolated, and we can now use them in applications.
And that's what you're doing in your lab. You're studying circadian rhythm. Tell us about it.
First, let me just say that one of the major applications of using luciferases has been as a way to assess expression of genes, what we call gene expression, when genes get turned on, when they get turned off. And by having a gene from another organism like the luciferases from bioluminescent creatures, we can then assess in another organism like human cells whether a gene's been turned on or off just by whether the cells start glowing or not. And so this is what we call making a transgenic organism. We take a gene from one organism and put it into a different organism. So we have been able to clone, many different laboratories have been able to clone the genes that encode these luciferase proteins that create light in the bioluminescent organisms. And then these genes have been able to be put into other organisms, such as human cells, under the control of genetic elements that allow us to know when these genes would normally get turned on and off. And so now they become a way of visualizing or reporting when genes get turned on and off. And so that's probably most familiar to people in the case of, for example, a tumor cell. When a cancer happens, some genes get turned on, some genes get turned off, and it's possible to put a luciferase gene into a cancer cell and see when the cell becomes cancerous, it turns on, and then the cell becomes luminescent. So that's kind of a turn-on that is it's kind of like a signposting or a spotlight in a certain kind of cell that this cell's gone bad, and we're using the luciferase as a reporter to see that as a label. But it turns out that gene expression is very dynamic, and genes are getting turned on and off all the time. There's a temporal aspect to that, such as in your stomach, genes are getting turned on when you eat a meal and things like that. And so it's also possible to use luciferases to see changes in gene expression, not just to turn on, but turning on and off. So that's how people that work on biological clocks, like me, have been able to really take advantage of the luciferases that bioluminescent organisms make. Because as the clock turns genes on and turns genes off, we can, by a number of genetic tricks, make it turn on and off the luciferase gene that we insert. And that way, the cells start to glow rhythmically.
And that dramatically changed how you were doing research. Before, you had to stay up at all hours of the day to get your research done?
That's absolutely right. Thank you for saying that, Lucy, because when I was a graduate student 30 years ago, if we wanted to measure biological clock phenomenon, I remember staying up for 3 days on a row and taking time points every 2 or 3 hours, and it was very grueling. And we could only do a few samples. Nowadays, with the luciferase reporter that comes from the bioluminescent organism, we can automate the system by using a very sensitive camera or a very sensitive light meter type apparatus. And we can be looking at thousands of cells, thousands of organisms at the same time simultaneously. So that's why it's been so useful in the case of looking at biological clocks. So let me just mention why biological clocks are important. So the technical term for them is circadian rhythms, and they are controlling our daily activities. They regulate our sleep. And there's a lot of different kinds of disruptions that happen in modern-day life, such as shift work or jet lag or a phenomenon called social jet lag. Social jet lag is something that's probably familiar to many of us that get up on time to go to work or school during the week, but on the weekends we sleep really late and then go to bed late, so that every Monday morning we're really kind of in a jet lag situation. So the term has been coined social jet lag. And these kind of disruptions put us at a significantly higher risk for obesity, diabetes, heart attacks, some kinds of cancers, gastrointestinal problems, and cause a lot of sleep disorders. And that's because we have this biological clock that's regulating our sleep, it's regulating our athletic performance, it's regulating our mental performance. And if we're not optimally phased with our daily cycle, then these kind of deleterious health effects occur. So the phenomenon is important for health and this sort of thing. How would we try to get it? Well, one of the things that scientists are trying to do is to try to identify different kind of pharmaceutical treatments that might help people that are shift workers or with jet lag or social jet lag. And that means screening hundreds of thousands of chemical compounds as potential drugs. So that's where the luciferase reporter comes in very handy. It would not have been possible 30 years ago to have done this because, again, people like me as a graduate student just can't do thousands and thousands and thousands of samples for three or four days continuously. So what's been possible to do is to take human cells, insert a luciferase gene coming from a bioluminescent organism such as a firefly, and have that luciferase controlled rhythmically by the biological clock so that we see these cells now become glowing rhythmically. We can assess how the clock is running by this bioluminescent reporter, and then we can dispense these cells out into many different kind of test tube equivalents and measure the glow of thousands of them at a given time, and so we can test hundreds of thousands of different drugs over the space of a few months, which is what a big pharmaceutical company or other kind of a chemical screening facility needs to do in order to assess whether certain compounds might act as drugs that might be useful in treating biological clock disorders.
I'm speaking with Dr. Carl Johnson, professor of biological sciences at Vanderbilt University. We're learning about how he uses bioluminescence research in his lab to study circadian rhythm. In studio with me, Dr. Mark Zimmer, professor of chemistry at Connecticut College in New London. We're talking about bioluminescence. There's also biofluorescence. You primarily study biofluorescent proteins. Tell us the difference between luminescence and fluorescence.
So luminescence, if you had a dark room and you had, for example, a firefly, you can obviously see it. If something's biofluorescent, you wouldn't see it. Fluorescence is when you shine a light on something, it absorbs that light and then gives you back a light of a different color with less energy. And typically, as Edie said right in the beginning, in the ocean, blue light travels the furthest. So most organisms that are fluorescent from the ocean, if you shine a blue light on them, they'll give you back a color like green. And so that's biofluorescence. We can't see it. We typically need glasses on, so we don't see the blue light because that would be too bright. But fish only see a small part of the colors that we do. And so they can actually see fluorescence. We, for example, can't see infrared or ultraviolet, but a mantis shrimp can see ultraviolet, infrared, and visible light. So we can all see different colors.
We heard Carl Johnson talking about the importance of this research, especially in medical advances. Talk us through when the green fluorescent protein was discovered. This was something that led to the 2008 Nobel Prize in Chemistry, a very big deal in the research and science community. What exactly is it and how it was discovered?
So it was discovered by a Japanese researcher, Osamu Shimomura, while he was at Princeton, who was trying to figure out how jellyfish, a certain type of jellyfish called Aquaria victoria, gives off light. And it gives off green pinpricks of light. And everybody thought it would be luciferase, which we've been hearing about, the most common way of making light. But he had to catch about 1 million jellyfish. It took him about 30 years. And he figured out that the jellyfish had something like luciferase that made blue light. But then there was something that sucked up all this blue light and immediately gave it off as green light. And it was called green fluorescent protein. And so he was just interested in figuring this out. He didn't want to use it or anything like that. There was another scientist, Doug Prasher. He thought, wow, we could take this gene, this recipe to make this green fluorescent protein, and we could stick it at the end of any other recipe. So you can think of it as having a recipe for a cake, and you sneak on the extra icing recipe. So whenever somebody makes a cake, there's a bit of extra icing. Here, whenever a protein's made, there's like a little light bulb at the end of it, and you can see where it goes. And he proposed to do this with cancer. So one of the problems with the cancerous growth, the tumor, is you actually need to cut it out, do a biopsy, look under a microscope, and then you can tell, is it a cancer or not? And in a mouse or in a person, you can't see if the cancer moves around. But if you made it fluorescent, so you shine a light on it and then it gave off green light, you could see where it moves. And so he proposed to do that. And he caught 70,000 jellyfish, found the recipe, the gene, how to make it, made it in bacteria, but it didn't glow. And he gave up, and not surprisingly, because if a protein is just a bunch of amino acids, and they have to fold in a certain way. You can think of it as a sentence. If you read a sentence, it doesn't magically fold into something and light up. And it's the same in proteins. There was no protein known that would fold itself and then light up. So Prasher gave up. Marty Shelfie at Columbia read this paper, had a student, and tried to do it himself with a student. But there'd been a new technique called PCR. They did it, and it lit up. And so now there were numerous uses. There's a company called Anti-Cancer Inc. that's using Prasher's original idea. They take human cancers, genetically modify them so they glow green, put them into a mouse, and then you can shine a blue light on this mouse, and you can see a single cancer cell anywhere in the mouse. So if it's a breast cancer, one cell breaks away, goes into the lymphatic system or the blood system. You can track this single cancer cell, see, does it stop and grow and cause a new tumor somewhere, or does it just die? And you can track all of this in real time. So it's become incredibly useful. And then there was somebody called Roger Chen who came up with the idea of, well, let's find different colors, red ones, blue ones. And so you can do this in two ways. You can go into a submersible and try and find fluorescent organisms that give off red fluorescence, or you can go into the lab and change the one you have to give different colors. And we now have a whole rainbow of colors.
So these green fluorescent proteins are a real powerful tool to see the inner workings of a cell.
Yeah, you can now see when something's made inside a living cell, where it moves to, and how much of it is made. You can also change the fluorescent protein so it only lights up under certain conditions. Edie Widder had spoken about bacteria that light up with certain toxins. You can now make fluorescent proteins that only light up with one toxin, DDT or something like that. Or you can make some that light up in the presence of calcium. When your neuron fires, your brain cells fire, there's an increase of calcium, a 100-fold increase. So you can now genetically modify a mouse, put this green fluorescent protein with a calcium sensor in the brain cells, and when the brain cells fire, they light up. So now you can see which brain cells are responsible for storing a memory, from taking a memory out, and what goes wrong. You can actually train a rat to go down a maze. When it sees a certain sign, it'll turn left. Sees a different sign, it turns right. You can see the brain flash when it remembers this, and then you can see it putting it in the wrong place in about 4% of the times. And then you know when it gets to the end of the maze, it's going to turn the wrong way. Something we never could see before and never knew about before.
I wanted to turn back to Dr. Carl Johnson, professor of biological sciences at Vanderbilt University. Can we talk about some of the future research that you're working on using bioluminescent sensors? We were hearing about green fluorescent proteins, but I know because you use primarily the luminescent proteins, how that's being used to learn more about brain cells.
Oh, well, thank you. Well, actually, Dr. Zimmer was doing a great job there, and I really appreciate him mentioning those things. And I also want to emphasize that the work that's going on in my lab, there's many laboratories that are taking advantage of these different approaches, and I think it's all really terrific. One of the things that we're doing in my lab that's not directly related to the biological clock is something that Dr. Zimmer also just mentioned, that is to create luminescent proteins that now respond to calcium, to do exactly what he was just talking about, to look at neural activity now in a very fast time span of millisecond signaling. So I mentioned before using luciferase reporters for gene expression, things that may take hours but are temporarily changing. But we're also trying to modify luciferase proteins to be sensitive to calcium or to voltage so that we can see electrical signaling in the brain. And I think this has tremendous potential because it can also be coupled with another phenomenon that's coming, which might be a great subject for another show, and that is optogenetics, which is using proteins that respond to light to actually open channels, and this is in the nervous system. And this is something that neurobiologists now are taking advantage of to actually control neural activity, and this is something that will probably also be clinically very relevant.
That's funny you brought that up, Carl. Mark Zimmer, that's your future book coming out soon on optogenetics.
I have a book
terrific
for young adults.
We should get together and talk sometime. But anyway, obviously we have a lot of very common interests, and so this is all a very hot area of research, both controlling neural activity and brain activity using light, but also recording it and monitoring using either luciferases or the green fluorescent protein that Dr. Zimmer was talking about. So I think all of this is just a terrific example of how studying really phenomenally interesting biological phenomena, as Dr. Widder was talking about, bioluminescence is just fascinating. How studying those phenomena, which not, at least at the beginning, weren't obviously of clinical importance, can be translated then into discoveries that help human health and medicine. And so the basic research aspect of all of these different things has been a tremendous benefit in terms of the kinds of things that we're going to be able to do clinically in human health down the road.
Carl Johnson is professor of biological sciences at Vanderbilt University. Thank you so much for joining us.
It was a pleasure. Thank you so much.
This is Where We Live. I'm Lucy Nalpathanchel. Dr. Mark Zimmer is here, too, a chemistry professor at Connecticut College, author of Illuminating Disease, An Introduction to Green Fluorescent Proteins. Today, we're learning about bioluminescence and biofluorescence. After the break, Dr. Zimmer will give us more examples of how the use of glowing proteins is advancing medical research in several fields. He'll also introduce us to Edgar. He glows, but not like Rudolph. We'll explain coming up. This is Where We Live. I'm Lucy Nalpathanchel. Today we're diving into science on the show. Have you heard about bioluminescence or biofluorescence before? You've seen it in nature. Think about the lightning bugs, the fireflies you've seen glowing in the summer night. Or if you vacationed off Vieques in Puerto Rico and have seen the bioluminescent bay glow due to tiny sea organisms making their own light. In studio with me is Dr. Mark Zimmer, professor of chemistry at Connecticut College in New London. And I wanted to actually take a call. Someone has a question about what I just mentioned about Vieques. Mary from Hebron. Mary, go ahead with your question or comment.
I have a question about how the bioluminescent bay or swamp survived, and if it did survive, the recent hurricanes and other environmental problems in Vieques, Puerto Rico.
That's a great question. Before we find out if they survived, what is it about the bioluminescent bay? What's glowing in there, Mark?
There are these tiny little diatoms. They're called dinoflagellates. They have two flagellate tails, two tails, and during the day they photosynthesize, but at night, if you touch their tail, they light up. In Puerto Rico, in the Bio Bay, there are about 700,000 to 1 million in a gallon of water. Touching them lights them up, so a fish comes in, it lights up around the fish, and then the fish gets a fright and will swim away. So it's a protective measure. There are about 8 to 10 biobays in the world. The number's decreasing pretty rapidly, mainly because tourists go. They wear a lot of bug spray, a lot of sunscreen, and then put their hands in the water. It's really pretty. You move your hands every time it touches. It's like a pinprick of light. And you can lift your hand, and if you look up in your arm, it'll be like stars in your arm because you can see these individual dinoflagellates lighting up. The reason they're in these bays is the temperature is exactly right. So they have to have a very narrow entrance, have to be about 4 to 10 feet, no deeper than that. And mangroves around help as well. So if any of this changed during the hurricane, then there would be a problem. If the entrance in the Mosquito Bay in Vieques is quite a long one, if that hasn't changed, then the bay is probably actually doing better now without all the tourists and will recover well. I know the bay in Fajardo has been doing really badly for the last 2 years, and this is probably just what it needed. Unfortunately, the people in Puerto Rico not, but...
We were talking about bioluminescence and how Carl from Vanderbilt is using it in his lab to understand circadian rhythms. But what are some other ways that bioluminescence has been used? In your book, Bioluminescence, Nature and Science at Work, you talk about ATP testing. Tell us what that is specifically and how it's helping researchers in their labs.
So ATP is your body's source of energy. Everything we eat at some point will get converted to ATP, and then we can use it for energy. Your brain gets its energy from ATP. We make probably about a quarter of our body weight of ATP every day. All living organisms have ATP. So it's really a way of figuring out, is something alive? If it makes ATP or has had ATP, it's alive. Firefly luciferase needs something called luciferin and ATP. So if you have no ATP, it won't glow. So you can now have a test for life. You can have like an earbud, and inside the earbud it's saturated with luciferase, and you push it along a surface. If there's anything that had ATP, it would glow. So Coca-Cola and abattoirs and people like this, they use this for a test. Are there any bacteria on the surface? Is the surface contaminated? Before this test, you used to have to actually take a swab, grow it on a petri dish. This took days. And then you'd know, oh yeah, it's contaminated. On the Mars rover, there was a luciferase test for ATP to see, is there life on Mars? It didn't light up.
Earlier, you talked a little bit about how the green fluorescent proteins being used in labs to advance medical research. There was something that I'd heard you speak about in a TED Talk about pregnancy and women that are at risk for heart attacks. That's really fascinating. Can you walk us through briefly?
So if somebody has a heart attack, the heart gets depleted of oxygen, the muscles die. And if the person survives the heart attack, those muscles never recover. In a zebrafish, they recover, but not in humans, except in pregnant women. Pregnant women have a heart attack, and it's more common than we would think, survive, they'll recover. And so what's happening here? Hina Chowdhury is a cardiologist at Mount Sinai. She had this crazy idea that if somebody has a heart attack, a distress message is sent out. Basically says to the body, help, I've had a heart attack. There's a molecule that's sent out, but there's nothing in the body that can help. In a pregnant woman, that message goes out, goes across the umbilical cord to the fetus. And now the fetus has embryonic stem cells, and they're released in response to this molecule. They go into the bloodstream, which will go to the heart, where this molecule, this distress signal comes from, and they'll fix the heart muscles. So how do you prove this? Hina had this really simple experiment. She took a female mouse, a normal female mouse, so nothing in the female mouse glows, and a male where every single cell is fluorescent, like Edgar, who we'll talk about later. Mate the two of them, and half the offspring will be fluorescent, and half won't. You can actually point a flashlight, a blue flashlight, at a female's stomach, and you will see like little jelly beans, half the offspring glowing. If she has a normal pregnancy, we can look at the female, there'll be nothing in the female glowing. However, if you induce a heart attack while she's pregnant, and she survives that heart attack, and then look for fluorescent, the only place you'll find the fluorescence is in the heart and in the heart muscles. And so the only way that could have possibly have happened is if these cells came from the embryo. And they didn't go anywhere else. They only went to the distressed muscles that needed to be fixed. Wow. And so there was a very, very simple experiment and a very, very cool idea.
Now, you mentioned Edgar. I don't want to run out of time. He is an example of a transgenic organism that produces GFP. Tell us who Edgar is.
He's an axolotl.
He is actually a salamander from Mexico?
Yes, they are two lakes outside Mexico City. There are probably 1,000 to 2,000 left in nature. So they're on the border of being naturally extinct, but in the labs, they're really easy to keep. It's a salamander that's stuck in its larval stage. So normally salamanders go from eggs to larvae to adults. Edgar is about 12 years old. He's a grumpy old man. But he's stuck in the larval stage. He's sexually mature. But what that means is also he's incredibly regenerative. If you were to cut off his leg, he'd be able to regrow it. If you cut off his optic cord, he'd be blind for a couple of weeks and then would be able to regrow his optic cord. So he's very interesting because of that. And so he's been genetically modified, so every single cell glows green. I just use him to take to high schools and schools and show kids. But you could, in theory, take stem cells from him, implant them into a salamander. If they differentiate and become something new, like cone cells in the eye or skin cells, if they came from Edgar, they'd glow green. And so you'd know where they came from.
Now, we talked about how valuable this is in modern biology. Challenges in getting research, especially from the federal government, to keep this kind of research going.
To me, the important thing is the basic research question that we've come to. Osamu Shimomura got funding to find out why the jellyfish gave off light. In today's climate, he probably wouldn't have got that money because there was no end result, no practical use for it. There are probably thousands of organisms in the oceans that have very, very useful proteins that we can use. For example, optogenetics, that protein comes from algae. And one can take these algae that respond to light, they use this to find out where the sun is, and put this into a brain cell, shine a blue light on the brain cell, and the brain cell will fire. Who knew that was an algae? And so we needed this basic research into algae to know this before we could actually use the proteins. The same with the fluorescent proteins.
Well, I want to thank Dr. Mark Zimmer again. He is a professor of chemistry at Connecticut College in New London. We'll look forward to your new book. Tell us again the title.
It's Lightening Up the Brain, the Science of Optogenetics.
I want to thank you for giving us a glimpse into, again, this very powerful tool being used in research in many labs. Thank you so much, Mark.
And thank you for having me here.
I'm Lucy Nalpathanchel. Thanks also to our technical producer, Chion Wolf. WNPR's executive producer is Katie Tolarski. Special thanks to Lydia Brown. As always, thanks for listening.